Welcome back to this next video, and in this video I want to focus on the active and passive transport, and I'll be giving you examples to help you understand the concept of the active and passive transport. Now the first thing is why this transport across the cell membrane that is necessary and why it matters. Now the semipermeable nature of the cell membrane, that is the phospholipid bilayer, that is very important for maintaining the delicate homeostasis of each cell. Now conditions in the body and in the cells, they must be constantly controlled because the cells depend on the body's environment to live and function. Now the maintenance of the conditions by homeostasis is very important because in the wrong body conditions, the important processes of the body or of the cells like the osmosis and the proteins, especially the enzymes, they will not function properly. And if you cannot maintain this homeostasis, if you cannot provide this optimum conditions to the osmosis or to the enzymes or the other processes, that is going to negatively affect the functions of the cells. Now what this means is that the transport across the cell membrane, that must be tightly controlled, because this is the transport across the cell membrane that help you maintain this homeostasis. Now the neuronal signaling, that is an important example where the movement of ions is controlled and used for the transfer of message from one neuron to another neuron. What I mean by this is that if you are not able to maintain a homeostasis of these ions, that means you cannot control the neuronal signaling, and if the neuronal signaling is not controlled, that is going to affect the body very negatively. Now, in this particular slide, I want to focus on the different types of the proteins or the protein channels and the different types of the transport that occurs in the body. Now, a protein that is involved in moving only one molecule across a membrane is called a uniport. And you can see over here, this is a uniporter or a uniport in which the protein is moving one molecule in one direction. The protein that move two molecules in the same direction across the membrane, they are called symports. They are also called as the synporters, the synports, or the symporters. And you can see over here that this particular protein that is actually moving two things in the same direction. If two molecules that are moved in the opposite direction across the bilayer, as you can see over here, the A is going to move inside the cell, the B is going to move out of the cell. That means that the two molecules that are moving in opposite direction, and this kind of the protein that is doing this particular phenomena, they are known as the antiports. Now the proteins which are involved in moving ions, they are usually called as the ionophores. Now, when you talk about the transfer of the molecule across the cell membrane, there are two major ways that molecules can be moved across a membrane. And the distinction has to do with whether or not energy or cell energy is used. One of the type of the transport that is known as the passive transport, and as you can see over here, no energy is used when you talk about the movement of the molecules across the cell membrane by the passive transport. But when you talk about the active transport, as you can see over here, ATP, that means energy that will be utilized for the movement of the molecule into or out of the cell. So in this particular video, I want to focus on the active transport, and in the following video, I'll be then focusing on the passive transport. When you talk about the active transport, it is actually the process of transferring substances into, out of, and between cells, but this particular transfer is by the use of energy. Now the cell often needs to transport material against their concentration gradient, and in these cases active transport is required. So this is one of the important things to keep in mind, is that in active transport you will be using energy and you will be moving things against their concentration gradients. Now active transport is most commonly accomplished by the transport protein that I just told you, And these transport protein, they actually undergo a change in shape when it binds with the sulfuse, a molecule known as the adenosine triphosphate, or the ATP. And this will be clear in a while. But what I mean by this is that an ATP molecule is going to bind to the transport protein. The transport protein is going to change its shape, and the change in the shape of this particular protein will be responsible for movement of the molecules against their concentration gradient in the active transport. When you talk about the types of the active transport, one of the important types of the active transport is known as the Antiport pumps. And as I've told you, the Antiport pumps, they are the membrane co-transporter proteins that pumps one substance in one direction while transporting another substance in the opposite direction. So you are moving two things, but they are in the opposite directions. Now these pumps, they are extremely efficient because many of them, they can use only one ATP molecule to fuel these two different tasks. You are doing actually two things. You are moving one thing out of the cell, you are moving another thing into the cell. That means you are performing two functions. But these pumps are efficient because they are using only a single ATP molecule to give you both of these functions. Now, one of the important type of the Antiport pumps, that is the sodium-potassium pump, and which is very important in the neuronal signaling. I have a detailed video on the neuronal signaling, how this sodium-potassium pump actually helps in the action potential, and I'll share the link in the description. When we talk about this sodium-potassium pump, as you can see over here, usually what happens is that outside of the cell, if this is the outside of the cell, usually the concentration of sodium is higher outside of the cell and it is low inside the cell. Similarly, when you talk about the potassium, the concentration of the potassium is high in the cell and it is low outside the cell. So what the sodium potassium pump do is that it is going to move the sodium from inside of the cell to the outside of the cell. That means you are moving against the concentration gradients. Now what happens is, if you can see over here, this is an ATP molecule. There is hydrolysis of the ATP molecule, and when there is hydrolysis of the ATP molecule, one phosphate group, it binds to this sodium potassium pump, and the ADP molecule that is released. Now when there is binding of this phosphate molecules, that actually helps in the attachment of these three sodium ions to the active site of this pump, and when they bind over here, the binding of this phosphate is actually going to change the shape of this particular sodium potassium pump, and the change in the potassium pump is going to move the sodium outside the cell against their concentration gradient. Now what happens is, when there is a change in the shape of the sodium potassium pump, the sodium is moving outside the cell, but at the same time it is providing binding site for the potassium. And as you can see over here, when the pump is in this particular conformation, it cannot bind potassium. But when it is in this particular conformation, because of the binding of the phosphate group, it is going to change shape. The potassium is going to bind to the active site of the sodium potassium pump, and the potassium is going to move inside the cell. So this is one of the example of the active transport in which you are using the energy of the ATP to drive the movement against the concentration gradient. Another important example of the active transport, that is known as the Antiport pumps. Now in case of the Antiport pump, a substance that want to move from an area of high concentration to an area of low concentration down its concentration gradient is used to carry another substance against its concentration gradient. Now one of the important example of the Antiport pump is the sodium glucose transport protein. Now, if you can see over here, you will be moving two things. One of the thing is moving against its concentration gradient, and one of the thing is moving down its concentration gradient. And one of the good example of the Antiport is the glucose reabsorption mechanisms. Now what happens is when you look at over here, you have got this sodium potassium pump over here in the wall of the proximal tubules, and what this sodium potassium ATPase do, that it is going to create an electrochemical gradient. What I mean by that is that this particular sodium potassium ATPase pump that we just discussed is going to move the sodium from inside of the cell to outside of the cell, and at the same time moving the potassium into the cell. What this creates is an electrochemical gradient. There is a low concentration of the sodium at this part of the proximal tubule, and the concentration of the sodium that is high over here. That means that the sodium is going to move down its concentration gradient. Here the concentration of the sodium is high in the lumen of the proximal tubule. The concentration of the sodium is low in the wall part of the proximal tubule, so the sodium is going to move down its concentration gradient. But at the same time, there is a high concentration of glucose in the wall as compared to the lumen, but this movement of the sodium ions also facilitate the movement of the glucose down its concentration gradient, because here the concentration of glucose is high, here the concentration of glucose is low, so the glucose is moving down its concentration gradient. And this movement is actually because of this electrochemical gradient that has been created by this sodium potassium ATPase pump. Now the protein or the protein molecule or the protein pump that is responsible for the movement of the sodium and the glucose into the wall part of the proximal tubule is actually known as the sodium dependent glucose transporter, or for short that is known as the SGLT. Now what happens is when the glucose move here the concentration of the glucose that is going to increase, and in the interstitial space the concentration of the glucose is low, so then the glucose is moving into the interstitial space by the passive diffusion, because it is moving from higher concentration to the low concentration, and this movement is actually carried on by another protein which is known as the GLUT, or which is known as the glucose transporter. So as you can see over here, two things they are moving. One is moving along its concentration gradient, the other one that is moving down its concentration gradient. So this will be an example of the Antiport. Another important type of the active transport that is known as the endocytosis. And the endocytosis is actually the process by which the cell internalize substances from their external environment. What this means is that the things they will be moving from outside of the cell into the inside of the cell. Now what happens during the endocytosis is, if you look at the steps of the endocytosis, during the first step, the plasma membrane that folds inwards, it makes an invagination, forming a cavity that is filled with the desired material. And you can see over here, if this is the desired material that you want to move into the cell, this actually fills in this invagination of the cell membrane. Now what happens is the plasma membrane then folds back on itself until the ends of the enfolded membrane that meet with each other, trapping the desired material. And when the desired material that is trapped, the vesicle is then pinched off from the membrane as the ends of the enfolds membrane diffuse together, and the internalized vesicle is then processed by the cell as per their requirements. Now there are three primary types of the endocytosis. You can see this process of the endocytosis in three different mechanisms in the body. One is known as the phagocytosis, the other one is known as the pinocytosis, and the third one is known as the receptor-mediated endocytosis. Now this phagocytosis, that is very important when you talk about the immune system of the human beings, and I'll have a detailed video on the phagocytosis. So this phagocytosis, which is also called as the cell eating, it actually involves the intake of the solid material, the food particles, or the pathogens. Another important one, the pinocytosis, which is also called as the cell drinking, that actually involves the intake of the molecule which are dissolved in fluid. So in phagocytosis, you are dealing with the solid material. In pinocytosis, you are actually moving the things from outside of the cell into inside of the cell, which are actually dissolved in the fluid. The receptor-mediated endocytosis that actually involve the intake of molecule based upon their interaction with the receptors on a cell surface. So there will be a receptor on the cell surface. The desired material is going to interact with that receptor, and then that receptor is actually helping in this process of the endocytosis. Another important type of the active transport that is known as the exocytosis, which is actually the opposite of the endocytosis, because endo mean inside, that means you are moving things from outside into inside of the cell. Exo mean out, so you are actually moving things out of the cell. So this is a process that is used to transport material from inside the cell to the external part of the cell by the use of energy, because we are talking about the active transport. Now, generally in exocytosis, a special vesicle which is bound to the cell membrane, it actually containing the cellular particles that will be expelled outside the cell. Now, this mechanism has been used in the removal of the waste material from the cell. It has been used in the transport of hormones and proteins and chemical signals between cells and the construction of the cell membrane. Now, the vesicle that is involved in the exocytosis, they are formed by the Golgi bodies. That means that the vesicle containing the desired material that will be made inside the cell, and these vesicle, they are formed by the Golgi bodies, by the endosomes, and by the presynaptic neuron, which are called is the neurotransmitter vesicles. Now, these vesicles, they fuse with the cell membrane, releasing the content outside the cell. As you can see over here, you will be making a vesicle inside the cell. That particular vesicle is then going to move towards the cell membrane. It is going to fuse with the cell membrane, and when they fuse over here, you will be having this pore-like structure, and in that particular pore, the desired material that will be removed from the cell into the outside environment. And one of the important example is the release of the neurotransmitter. This is the sending neuron, which is also known as the presynaptic neuron. This one is the receiving neuron, also known as the postsynaptic neuron. And as you can see over here, these neurotransmitters, they are made by the cell. They are then packed into this particular vesicle. As you can see over here, this will be packed in these particular vesicles. These vesicles are then going to move towards the presynaptic membrane, and when they move towards the presynaptic membrane, they are going to fuse with the presynaptic membrane, and when they fuse over here, the neurotransmitter that will be released into this area, which is known as the synaptic cleft. When the neurotransmitter they are released, there are special receptors on this receiving or the postsynaptic neuron, and when these neurotransmitters they interact with these receptors, a cellular response that will be generated. So these are the four important types of the active transport. Now, one of the important thing to understand is the difference between the primary active transport and the secondary active transport. Both of them, they are active transports, but the active transport is further subdivided into these two types, the primary active transport and the secondary active transport. As you can see over here, in the primary active transport, you will be using the energy, the ATP as energy, but in the secondary active transport, the protein molecule that is involved in the active transport, that is not directly using ATP, but it is actually using the electrochemical gradient for the movement of the molecules against their concentration gradient. And one of the important example of the secondary active transport that we just discussed was the transfer of the glucose from the nephron back into the blood, where it is absorbed by the different cells of the body. So in the next video, I'll be talking about the passive transport and its different types. So if you like the video, please subscribe to my channel, hit the like button and share it with your friends.